Hello ecology class, my name is Lorne Green and today I'd like to discuss my microcosm project titled To Live and Let Die in My Microcosm. An overview of this project was built in order to learn more about the energy inputs and outputs inside a biosphere similar to what we live on here on Earth. Project shows us a delicate balance between inputs, storage, and outputs here on Earth and what we as humans can do to alter those relationships. I feel like this microcosm project helped me to understand how all living things are interdependent on each other in one way or another. And we all are affected by changes or alterations to our environment. The small-scale experiments like these microcosms can be of great benefit when tackling global issues like climate change and better understanding of how to manage our natural resources here on Earth. This project was designed as an aquatic environment and sealed 7 weeks ago with no other introductions into it other than energy from ultraviolet light. It has remained on my windowsill on the south side of my home for that duration. My original hypothesis, which was included in our first homework assignment, was I think the microcosm will turn into a green tinted jar with everything turning a more green and vibrant color. I think the reasons for this is the controlled temperature of my house will allow conditions inside the jar to mimic that of a tropical rainforest. Also, the warmth of the sun coming in through the window and the refraction of light through the jar will also warm the temps inside to a level that will allow the algae and other sun-dependent organisms to flourish in such a small environment. Here you can see a map showing the location at which I collected this microcosm contents. You can see right here, if I can find my mouse, right there is the county in which I live, and it's where I actually did collect the materials for this experiment. It's a slough along Point Remove Creek, just on the western side of this county. One thing to take note is it's been a lot of flooding in this area, and almost all water is extremely high, and I think that affected my contents. I was not able to get out there to exactly where all the aquatic plants are normally living, so my collection was probably in a site that might dry up during the dry months of the summer, so that could affect the success of my microcosm. In this slide you can see where I collected the materials for my experiment. On the left side you can see this is me right here looking for a good spot. In the photo to the right, that's me where I found some good materials, and I basically put 2 or 3 shovelfuls of organic matter and soil into the bucket, and I don't know, maybe 2 gallons of water, and took that back to the truck. And from this, I filled my containers up with the soil, organic matter, and water. And I did collect a couple of plants. I think one was a cattail and the other was some duckweed. Here you can see the containers I used. It's basically 2 orange juice containers, basically all plastic. Approximately 1,474 grams in each container. That turns into about 1,306 cubic centimeters. I tried to fill them up about 1/3 of solid material with some organic matter and then 2/3 water. I did put a couple of plants in each, 1 cattail and some duckweed that I found in each container. Did not put any heterotroph organisms in there. I felt like there would be enough just from the organic matter, but later we'll see whether or not that was true. This slide shows the containers from day 1 to the very last day of this experiment, which was basically 47 days. During that time, About 22 days were considered what I call sunny days, which are about half of the time. Then the other half was cloudy days, and then about 5 days that were partly cloudy. There was a lot of rain during this time period. I probably would say that during the first 2 or 3 weeks, they were receiving 5 to 6 hours of direct sun. And then the last 4 weeks, based on foliage growing on a couple of oak trees in my backyard and the movement of the sun angle, I was not getting as near as much sun in the window. So I'm going to say somewhere between 1 and 3 hours of direct sunlight for 4 weeks. And that was primarily morning sun. Anything after, say, 11:30, there was not receiving any direct sunlight at all. This shows the weather patterns for March. You can take a look at this close if you'd like, but it'll just show the days that were sunny, cloudy, which days we received rain, the average low and high, wind direction, and wind speed, and the humidity. The data in the green box is representative of the yellow bars that's highlighted, so that's just showing that particular time frame, that particular hour on that day of what the temperature high and low was. This slide shows the weather patterns for April for Morrilton. Again, like I said, it's going to show that about 22 days during this time period with sunny days, 20 cloudy days with 5 partly cloudy days. Again, like I stated before, about 5 or 6 hours of sunlight during the first 3 weeks, and then about 1 to 3 hours of direct sunlight for that remaining 4 weeks. Here you can see 4 observations throughout that 47 days. This slide, right, this picture here on the far left is day 1, and this here is day 47. You can see a vast difference in the 2 photos, and the one thing that I did take note of was this airspace on March 8th had actually increased from this airspace here on March 8th. This was March 18th. Sorry, I couldn't see that 1 right there. But I had larger airspace during the 18th slide, and then on the April 5th slide, almost all airspace was missing. And then if you look at this last slide, you can see the airspace has come back. In this particular jar, this one here is more airspace than in this one, but I found that interesting, the gas exchange during this process, and you can see on the last 2 photos there how the jars are leaning. They become misshapen and warped due to pressurized interiors. I'm assuming through the gas exchange, the decomposition of the organic matter reacting with the water and the air. During my observations, the first week I observed the clouds and sediment settling out and the water becoming surprisingly clear, still with about 5 centimeters of airspace. Week 2, the water actually clouded up again. Airspace looked to be increasing to more than 5 centimeters. Bottles felt like they were getting pressurized from the inside. They felt tight when handling. The cattails were trying to grow. Green leaves were trying to get up into the airspace. By week 3, I noticed 5 centimeters of airspace had disappeared, like I mentioned earlier. There was also evidence of algae growing on the sides of the bottles, and gas exchange, again, was causing extreme misshaped forms of the bottles. They were starting to lean. You could see that in the photos. By week 5, there was algae growing all over everything. Everything looked fuzzy. The plants, the cattails and the duckweed looked dead. The water had turned dingy brown. And again, bottles felt very tight. The pressure seemed to be getting higher and misshaping the plastic, pushing out the bottom actually of each jug, causing it to lean. In the last week, the water became very dingy. You could barely see anything inside, the plants, organic matter, anything. The airspace had increased, and the pressure was at an all-time high, obviously. The major findings of this experiment show me that algae will flourish, the green plants needed much more airspace, and plastic is not a good container. During week 7, the water settled, got very clear, then got murky again, lost all airspace, and then gained it back. The algae took hold about week 5, covered everything. My plants died. The plastic bottles expanded and became so misshapen that they're leaning. I would say that my observations support my hypothesis, other than the plants not living. I predicted everything would get covered with green algae. That's exactly what happened. The one difference being is the algae is more brown than it is green. The 3 ecology concepts that I found that this experiment matched was first the ecosystem concept, the system of biotic and abiotic organisms working together and sharing the same space, made up of autotrophs, heterotrophs, and non-living organic matter. Not so sure about the heterotrophs. If I have any in there, I've not found them. Again, I think that might go back to where I collected my material. The food chain concept is the second concept. It's a sequence of where organisms feed and live off of each other. The sunlight is the energy source. The algae is feeding off of the decaying organic matter, as well as the gases produced during the decaying process. The niche concept, I think, would be the third. The algae seems to be the only thing growing in this microcosm, and it derived from a wetland community where I collected it. I don't think this would survive in a place, say, out in the desert or 100 meters under the surface of the ocean. In further research, I would hypothesize that I would like to test this to continue to watch the microcosm to see if any heterotrophs might appear, or to see if I added 30% more airspace, would the plants revive and begin to grow again? I don't know. Things I'd like to test. There are several things I would do to improve this experiment. First, I would switch the container from plastic to glass, probably increase it by double. Second, I would have 50% airspace by volume in my container, whatever container it was. Third, I would work harder when collecting the materials to make sure I get them from the heart of the wetland. The expanded edges of that flooded water kept me from gathering a representative sample, in my opinion. My microcosm in context, there is nothing simple about the environment in which we live. In fact, all the systems at play within our environment is so complex, in most instances, we can only theorize about what's going on. Microcosm study helps scientists to try to mimic what is happening in a particular ecosystem and try to predict outcomes from differing energy inputs. By studying the flows within the system, we can predict what might happen if we begin to have a drastic change in those inputs. Take climate change, for example. We can predict what might happen when solar radiation increases the temperature by 4 or maybe 40 degrees in a given ecosystem. There you can see my references, basically everything in our textbook, and then my weather data I got from timeanddate.com, the maps I made myself, the photographs I made myself. Thank you guys for your interest in my microcosm. Good luck to you in your future education.